Good morning, good afternoon, and good evening, everyone, and welcome to this Scientist.com and PhenoVista webinar titled Using In Vitro Assays to Model Complex Biology with PhenoVista's Assay Services. I'm Liam Sanyo from the events team here at Scientist.com, and I'll be your host today. We're being joined by Dr. Anna Wang, Product Manager at PhenoVista, and Adele DeSutter, Associate Director at Supplier Relations at Scientist.com. And first, Anna will discuss PhenoVista's development of complex in vitro studies to provide actionable and relevant data that can help drive drug discovery forward. Following that, Adele will give a brief demo of PhenoVista's digital storefront. And with that, I'm very pleased to welcome the first speaker, Dr. Anna Wang. And Anna, thanks so much for joining us today, and the floor is yours whenever you're ready.
Thanks for that introduction, Liam. Hi, everyone. My name's Anna. I'm the product manager at PhenoVista Biosciences. Thanks for joining today. I'm excited to share with you a few different types of assays and projects that we run at PhenoVista, working with various clients. So today I'll be going over how PhenoVista uses and develops in vitro assays to model complex biologies of various diseases in a lot of our drug discovery campaigns working with our clients. I'll start with a brief introduction in PhenoVista for those that haven't heard of us, and then I'll dive a little bit deeper here into our ready-to-go assay services and custom assay services and show examples of each of these. And I'll end with a brief discussion about explaining what cell painting is, as well as a case study there of a cell painting assay we ran in some hepatocytes before. So I'll start with a brief introduction for those of you who haven't heard of PhenoVista before. We are a US-based CRO. Our scientists, our team is comprised of scientists from all different backgrounds of biology, and we provide a collaborative experience with our clients and act like an extension of our clients' labs to help drive forward their research campaigns and drive their therapeutic candidates into the market, into the clinic, by combining our expertise in modeling complex biology with relevant cell models to provide rich data sets for our clients to inform their go, no-go decisions that are important to bring forward their research in the process of drug discovery and development. So overall, we pride ourselves on our collaborative nature of our engagements with our clients. We use cell models that don't just utilize simple cell lines, but we really have a lot of experience with using relevant cell types like primary cells, iPSC-derived cells. And we work with any sorts of cells or reagents that are commercially available or that clients might have developed in-house and want to provide to us to use in these experiments. We have specialized data in bioinformatics pipelines, and we custom design algorithms of our analyses to specifically answer the questions that our clients have that are specific and unique to their research goals. And like I mentioned before, we're very collaborative, so we do a lot of check-ins with our clients as the projects progress. We act as an extension of your lab to expand your bandwidth and help you with your efficiency in your research goals. A lot of companies that work with us are virtual, so maybe smaller or newer companies that don't have the resources yet or the space yet to have their own labs. We'll do a lot of work to help them get and gather new data for them to drive forward their goals. We also have worked with reagent providers that want to test their new products in new hands. So a lot of different types of clients come to us, primarily a lot of drug discovery and pharmaceutical biotech companies, but we do span a lot of different areas. In general, we work in three major therapeutic areas. So that's neurobiology, oncology, fibrosis, if we need to categorize them in three large bins. The vast majority of our projects fall under the CNS neurobiology space. I'd say probably about half, maybe even more than half of our 10-year history has been in this space and developing in vitro assays for various disease models and screening assays in the CNS space. And more recently, we've gotten in the last few years a lot of asks from our clients for assays in fibrosis. So we've expanded a lot of our offerings there, which I'll be delving into a little bit there. But I want to emphasize that we're not limited to these three therapeutic areas. They just seem to be the most popular ones that we get asked for. We've also worked in other areas like agricultural sciences, where Some companies have come to us before to test maybe the neurotoxicity of a new pesticide that they want to submit to the EPA approval, or maybe companies that work in animal health, so helping to develop pharmaceuticals for animals and pets. Alternative meat companies have come to us, maybe helping to figure out new culture conditions to help enrich for a certain type of nutrient. So a lot of the work that we've done has also fallen outside of these three spaces. Today, I'll be talking mostly about these three spaces and giving you examples within each of these. So our expertise is pretty broad. We like to say that our expertise in a nutshell is the biology. So developing the models to help answer questions in biology. The top row there are kind of the questions that our clients ask for or need answers to the most. So questions about, is my therapeutic efficacious in this specific model? Does it activate a certain pathway? Where does it go? Does it induce changes in the location of a specific protein of interest? Is it even taken up by the cells? So these are the kind of questions that we get the most often from our clients. And we take these questions and we work with them and dig through literature and use our own expertise and experience to develop in vitro models with different types of cells in different formats to create a system that is tailor-made to answer those questions for our clients. And this can encompass 2D models, 3D models, spheroids, organoids. We also have an in-house bioprinter that we use for some more complex experiments, cultures of single cells or multi cell types and within one culture. So it's a very flexible system that we provide our clients, and it's very much catered to what they need. And again, we like to use the most relevant cell type. So primarily all of our asks have been using primary cells or iPSC-derived cells. In-house, we specialize in high content imaging, so answering a lot of questions through phenotypic screening, phenotypic analyses. We also have a flipper in-house for measuring calcium flux in experiments that might be interested in the function of cardiomyocytes or like neuronal calcium flux. But we do have partnerships with other companies for our clients that also want to supplement their phenotypic or flipper data with data like transcriptomics, genomics, proteomics. So we kind of try to make this whole experience like a one-stop shop for our clients to get the most out of a project. And one of those actually is our new partnership with Neuroservices Alliance that's just one building down from us. They're a CRO that specializes in CNS electrophysiology. They provide functional readouts mostly in neuropharmacology, and we overlap in some spaces, especially like the neurodegenerative spaces. So clients that study Parkinson's, Alzheimer's. So we are constantly working with other partners to keep expanding our offerings and offer richer data sets so that our clients have better chance to understand how and what their test articles are doing. We can bin our service offerings into three main areas: image analysis, ready-to-go assays, and our custom assays. So the simplest would be the imaging analysis assays because it is the most cost-effective, and essentially you can ship us your stained fixed plates after having run your experiments in-house, and we will scan them on our microscopes and do the image analysis and give you quantitative readouts from that analysis. So that one is kind of the downstream, the last step of that we use in our ready-to-go assays and our custom assays. So moving up in complexity, the next one is our ready-to-go assays. This is our pre-configured assays, pre-validated, already optimized assay windows, and it spans a lot of different spaces in biology. So it's kind of a plug and chug version of our assay services. So clients can ship us their test articles, tell us which ready-to-go assay they want their test articles in. It is our most cost-effective because it is pre-validated, so we don't have to go through optimization and spend the cost and resources to actually design an assay. And it's the fastest full-service turnaround. So from receipt of your test article to delivery of a presentation ready report, on average takes about 4 to 6 weeks. The most complex and the most popular service that we have is the full service of our custom assays or bespoke assays. This is our most collaborative model. We essentially start from scratch. Our clients come to us with a question, maybe some suggestions or ideas, or maybe some experiments that they've run themselves in-house. And then we work with them to create a new model or repeat a model that maybe they just want to verify. It's very flexible. And this is where we get the most get to develop the most complex models. So those 3D models, bioprinted models, microfluidic formats. And it's fun to kind of work in between these different types of categories as well. So like we've had some clients that started with ready to go assays, noticed something interesting that the chest article did, and then took that data and moved it into a custom assay to delve deeper to answer questions that the ready to go assays may have brought up. So the benefit of working with us really is that you get all of these, it's all the same team. You'll have the same project lead on your project that will take your questions and understand what you really understand the journey of how you got to that question to develop new models or repeat or adjust experiments accordingly to really get your answers that you're looking for in your research. So first, I'll delve into some examples of our pre-configured ready-to-go assay services. We do have a long list of them, but I'll go over our 3 most requested ones, or the ones we get the most requests or questions about. These were chosen because over the 10-year history of PhenoVista, we noticed that a lot of clients kind of had the same types of questions. So we like to refer to it as the PhenoVista's greatest hits assay set. Like I said before, it's the quickest and most cost efficient, really quick turnaround time, especially compared to our custom assays. And they answer questions across a wide range of areas from neurobiology to cell painting, oncology, fibrosis, and they're very expandable. That we already have everything pre-configured, so you can ship us 1 or 2 TAs to hundreds of TAs over months, and we can just plug them into our system and get you data back in a really efficient way. And this kind of shows you our the variety of areas where these assays can be applied in. So across the top here, we have the different areas that some of our clients have used these assays in. Down the column on the left are a list of the actual assays. And when you go on our website, you'll be able to find little handout downloadables, one-pagers about each of these assays that explain the benefits of them, the types of readouts you'll get, and the kinds of questions that they can help answer. So from here, I'll just be going over a few select ones. Feel free to go to our website and look at any of these other ones that might catch your attention. But one of the most requested ones that we've gotten is our ready-to-go lung fibrosis assay. And you'll notice right under it is our liver fibrosis assay. They are essentially the same kind of workflow, obviously with a little bit different conditions and different cell types. But most of the things that I'm saying about that I'll be going over with the lung fibrosis assay can also be applied to our liver fibrosis assay. Those that aren't in the field might not be too familiar with fibrosis is. It's a disease in which the repair mechanisms in the tissues go into overdrive and overcompensate after repeated damages or insults to that tissue. And this will end in, this can end in a lot of deposition of extracellular matrix proteins and eventually lead to stiffening of the tissue to the point where the organ doesn't really function anymore and it fails and can lead to death. But this can occur not just in lung, in multiple tissue types. So our ready-to-go services cover lung and liver, but we have done assays, custom assays, where we've used kidney, dermal, and done very similar types of experiments, just tweaking conditions here and there. And although fibrosis itself is considered a disease on its own, it's also being more studied in terms of oncology, as we know that a fibrous layer of tissue often encompasses tumors, and that makes it really difficult for certain therapeutics to penetrate into the actual malignant tissue because of the fibrotic layer. So this assay is it gives you multivariate readouts. So a lot of different types of metrics that are hallmarks of disease, that are hallmarks of fibrosis. And because it's imaging, you get really clean readouts about those measurements that I'll show you a couple examples of the readouts that you get now. So with each of our assays and reports, you'll have a set of representative images. So that's what you see here on the left. In the case of our ready-to-go lung fibrosis assay, we use primary cells or human lung fibroblasts. And we've developed a protocol where we keep the fibrotic induction baseline really, really low. So we get really good measurements of induction of fibrosis. And we induce fibrosis by treating the TGF-beta. Our assays all include controls, and this assay will have a control inhibitor that you can compare your test article to. And here our staining palette is hooked for the nuclei, alpha SMA, which is a marker of stress fiber formation. Alpha SMA gets incorporated into these stress fibers, so we can quantify that. We also stain with phalloidin for total F-actin content. And then we quantify our images with a number of different metrics. Here on the right, you'll see one of the readouts, which is just fiber total area. And you'll see a dose-dependent increase of the stress fiber area with TGF-beta treatment. We also quantify separately the F-actin and alpha SMA readouts, since they're in two different channels. We see generally the same trends usually. And we'll not only be able to see total area, average area, intensity of staining in each well, because it's imaging, we can look at really detailed readouts like fiber alignment. So as the fibrosis is induced in these cells, we see more and more parallel fibers, which makes sense since it's what contributes to that stiffening of the tissue in the disease. And you can see here in the quantification that with TGF-beta treatment, there's a dose-dependent increase in that alignment of the fibers, which is prevented when we treat with a control inhibitor or test article inhibitor to prevent that from happening. Last year, we also added in a new feature that's optional for some clients that wanted it, which is fibronectin deposition. So we can take those wells, decellularize, and then stain and quantify for fibronectin content in those same cells and those same wells. So you get really parallel readouts here of all these different metrics. And we have done a lot of fibrosis work. It all started all of these studies started with our custom assays. So other areas that we've worked in here aren't just these readouts, but also things like actually look at pathway activation. So translocation of the phosphosmad into the nucleus. Those are studies that we've done quite often as well. And like I mentioned before, we have done this in other biological systems as well, dermal, kidney fibroblasts, and with multiple other readouts. So with our ready-to-go assays, you'll notice that a lot of them are in the CNS space since we've done so much work with them. Our most popular one in this area would be neurite outgrowth or neurite network dynamics. They're very similar assays. I'll go over the neurite outgrowth one first. So neurite outgrowth, really important to the development of new neurons, making sure that that development establishing that neurite network is super important to the functionality of your networks and the health of the neurons. So this and the network dynamics assay services are really popular for people that want to see if their CNS targeting therapeutic has some sort of effect on overall health. And looking at the neurites is a key way that a lot of our clients like to determine whether or not their TA is going to have an effect on that health of the neuron. So in this assay, it's very quick. We seed our neurons and then immediately within a couple hours, we'll treat with the test articles and compounds because the neurites grow pretty quickly. For our clients that are more interested in how their therapeutic might be affecting already established neurite networks, we have a very similar assay on a different time scale. So that's the neurite network dynamics assay. And then the difference here is that for the neurite network dynamics assay, we pre-culture the neurons for 7 days. So those neurite networks are already established before treatment. So you get different answers with a very similar workflow here. Our palette in both of those assays is really simple. It's hooks for the nuclei and then cell mask, which stains for the entire cell, so the entire neuron. And we have, in that assay, we include our controls, our staurosporine and vincristine as our controls. You can see on the right here with staurosporine, you get an increase in neurite outgrowth, whereas vincristine inhibits that. So you can compare your test article relative to these two controls. And we get a lot of different types of physical metrics here by using our algorithm to look at different aspects of our cell mask staining. In this graph here, you'll just look at, this looks at average neurite length. We also look at neurite area, and we can look at more complex things like branch points. So how many branch points do we usually see per neuron or per well with each of these treatments? We can also look at how far do those branch points start away from the cell body. So you'll get a lot of different metrics here with this really simple two stain palette that answers a lot of questions that can apply to basically any area of research that has to do with neurons. So anything with the CNS. The last ready to go I'll go over is the microglia phagocytosis assay. And this one is a live cell imaging assay. So the microglia are becoming increasingly implicated as big players in onset or progression of various neurological diseases. Microglia are kind of the helper cells, are kind of known as the immune cells of the CNS because they help maintain neuronal health. They gobble up debris and dead cells. They help prune synapses that aren't used just to keep everything neat and tidy. And so this phagocytosis function of microglia is really important to their function and to maintaining overall health in the CNS. So this is what this ready to go assay aims to provide answers to. So in this case, we do have wild type neurons, or we have a selection of Alzheimer's disease mutants. So you can compare the behavior of the microglia in these different lines, and how your test article might affect mutant versus a healthy wild type one differently in terms of their ability to gobble up what we use are fluorescent bacterial bioparticles. So this is live cell monitoring. We'll do a time course here. So you're able to look at both the rates of the phagocytosis as well as the maximum capacity of the microglia to eat up these bioparticles. And this one's interesting because it's a really simple one that you can take into more complex bespoke projects. So some follow-up assays might include, once you understand in monoculture with microglia how they're behaving, you may take it into a co-culture with neurons, for example, to look at how the presence of microglia might affect the different readouts in your neurite networks, which are projects that we've done before. But focusing just on our ready to go assay here, you'll see our representative images are kind of just quick shots from our time course over 24 hours. Down the column, down the side here, you'll have the different control compounds that we'll include in this assay. So you'll see that interferon gamma and cytochalasin D are very good at inhibiting this phagocytosis activity, whereas IL-10 increases it relative vehicle control. And as I mentioned, this is also, we do also have available some TREM2 mutants. So those that are interested in Alzheimer's and understanding the differences in wild type versus mutant microglia derived from Alzheimer's patients. So we have a lot of custom projects that have looked at other control compounds as well. So a lot of people are interested in how LPS affects this activity. And this is just a starting point for a lot of things that you can take into with our custom work, which I'll get into next. So the ready to go is kind of encompass our greatest hits. So those are ones that we've gotten a lot of asks for. But the majority of our business and our clients at PhenoVista come to us with very specific, difficult questions. And that's why we've kind of established ourselves really with our custom assay services. So developing assays that will specifically answer what they need. So I've just chosen a few different examples here. And these are also kind of our most popular asks, although because they are custom, every single project is very different. But in the CNS space, once again, one of the most popular ones we get asked about to perform is the synaptogenesis assay. So synapses are fundamental to communication between neurons. Healthy synapses are what allow neurons to talk to each other and to carry out their functions, both resulting in physical, cognitive, emotional functions, and in our brains, and also dysfunction in synapses or aberrant physiology in synapses is associated with neurodevelopmental disorders and neurodegenerative disorders. So a lot of people are interested in understanding how their test article might be able to maybe restore synaptogenesis or increase it, or if they're just curious overall on if their test article has some sort of detrimental effect on the health of the neurons through measuring a number of synapses. So in this specific assay I'll go over, we have a test article that we're testing for its effect on synaptogenesis. And this is in a co-culture of iPSC-derived glutoneurons and astrocytes. This case did have 2 different staining palettes. I'll only show data from the second one because it's the one that's related to synaptogenesis. But palette 1 would be, because it's a co-culture, you have Hoechst, which is the nuclear marker, TUJ1, which stains for all the neurites. And then we have a neuronal marker and an astrocyte marker. So we're able to distinguish between the 2 different cell types that are being co-cultured together. And then palette 2 is the synaptogenesis palette. So we'll stain for synaptophysin, which is a presynaptic marker, and PSD95, which is a postsynaptic marker. So detecting a synapse would be, you would want both the presynapse and postsynapse markers. So our overall goal here is to look for a co-localization of those 2 biomarkers in order to determine whether or not we can categorize it as a synapse. The setup for this experiment, synaptogenesis it takes a while. So these cells need to be cultured typically for most vendors at least 20, 21 days. The longer, usually the better, the more synapses you'll get, the better established these networks are. And in this case, we took it all the way out to 28 days, but we had 2 different time points. After culturing the cells for 20 days, we treated one set of the cells with the test article for 24 hours before fixing and staining on day 21. And then the second set, we carried it out for a week longer. So the final time point being at day 28. Here are some example images of those 2 different time points, these representative images. Again, these are co-cultures of neurons and astrocytes. So on the top row, you'll have day 21 cultures. The bottom row, you'll have the day 28 cultures. And here's our palette 2, so our synaptogenesis palette. Hoechst for nuclei. The second column would be our neurites. And then the 2 PSD95, synaptophysin, the post and pre-synaptic markers. And then we have blowups here on the far right to really zoom in on what we're working with. So this is the level of imaging and quality images and quality cultures that we need to be able to assess synapse formation and quantify how many synapses we are getting with these different conditions. So because, as you can tell from the bottom panel on the right, we have much more established neurite networks. There's a lot more neurites there. There's a lot more going on. So that's what we focused on for quantification. So here we have our representative images of our cultures that are treated with either vehicle, so DMSO, or 3 different concentrations, increasing concentrations of the client's test article to see what if this test article might be detrimental to synaptogenesis. And at the bottom, you'll have the blown up insets, so you'll be able to see the difference. You see that with 10 nanomolar TA versus 30 versus 100, you'll see even different just concentrations of the neurites and the staining intensities. And when we put this through our specially designed algorithm to look for co-localization of our PSD95s and synaptophysin to determine synapse counts, we can get quantitative data like here. So we did test different seeding densities with co-cultures. You want to test a few different conditions to figure out, ratios and what would be a good density of cells for us to still be able to measure and count clearly those synapses and be able to distinguish between neurites and cell bodies. So overall here, we have plotted the total number of synapses per well for each of these cell seeding densities. And again, they were identified by looking for co-localization of those 2 markers. And in general, you can see with increasing TA concentration, you don't see much of a difference with the number of synapse counts, with the number of synapses per well. So it looks like the TA here was not detrimental to synapse formation, which is probably what the client was hoping for. And overall, what you can see is that day 28 versus day 21, we do see higher counts of synapses with those more established cultures as expected. So we can take a very similar approach. So looking at co-localization of markers to look at other types of synapses. One of the most studied ones that we also get a lot of requests about are NMJ formation assays. So we can culture motor neurons from healthy or disease genetic backgrounds with myoblasts. And then we can look for acetylcholine receptor clustering. So co-localization of those or clustering of those proteins to determine formation of those junctions. So a lot of these assays, we have a similar approach, but by changing out the timing, changing out the types of cells and the culture conditions or what we're staining for, we can answer a lot of different kinds of biological questions. So kind of shifting gears a bit away from CNS, we do have a number of oncology projects that we have performed in the past, and I'll take this into a 3D assay as well to showcase some of those capabilities. So one of them that we get asked about a lot is tumor spheroid killing. Over the last decade or so, taking and modulating a patient's own immune system to fight their own cancer cells has been inspiring a lot of different approaches and new ideas for how to approach cancer therapies. In this case, we're using a 3D model, a pretty simple 3D model, but the idea is that this model might better mimic a tumor than a 2D model because it actually has more of the overall morphological similarities. 3D tumors don't really grow in flat sheets. But these are also asked for a lot, these 3D models, because you can answer questions with imaging that you might not be able to with other types of assays. For instance, one of them could be how deeply a therapeutic candidate can penetrate into a tumor. So how effectively can it go into those, into the tumor and access those cells to have more effective killing. In this case, we have a client's test article, which is a compound, and they wanted to see its ability to enhance the killing ability of PBMCs towards a tumor spheroid. So in this case, again, it was pretty simple. Compared to other projects that we've done, it was A375 cells that were grown into spheroids and then co-cultured at the PBMCs. To distinguish between them, our palette, we have hooks for nuclei, so the general marker. Cell tracker green was used to label the PBMCs, so our effector cells. And then cell tracker red was used to label our A375 spheroids, so our target cells. And then our stain, our readout, was DRAC7, which stains nuclei of dead cells. So for us to be able to quantify how many cells were killed. The method here is you culture the cells and you grow those spheroids first. We then pre-labeled our, with cell tracker, green or red, the spheroids and the PBMCs before mixing them together. Also before mixing them together, we stimulated the PBMCs with the client TA to try to see if that would help increase their killing potential. And then after 48 hours of co-culture of these two cells together, we fixed and stained for quantification. And here's a snippet of some of what we saw. Across the top, you'll see what you're looking at. So hooks would be the nuclei, A375s, that would be your spheroid of your target cells. In red, you'll have the PBMCs, so these are effector cells. And just by eye, you can see the decrease in the size of that spheroid with the addition of the PBMCs. Staurosporine here was our control because we know that it kills cells. So effectively, you can see that the spheroid has shrunk with staurosporine treatment. And also you see that pre-incubation of our client's test article with the PBMCs also enhanced this killing ability and led to smaller spheroids. And we can quantify all of this. There are a lot of different measures we can do here. I'm just showcasing two of those aspects. We're looking at a total spheroid volume of the A375s, the target cells, and the total surface area. So you'll see that with the increasing dose of our client's TAs, we saw increased killing of those spheroids. And then with staurosporine, it's pretty comparable to staurosporine with the higher doses of our client's test article. So in this case, this test article did promote the T-cell killing relative to vehicle control in a dose-dependent manner. And again, this one was with a compound to stimulate our PBMCs. We have also had projects and clients where they come to us with CAR Ts that they're designing. So instead of using PBMCs that are being stimulated, you whole culture a spheroid with the CAR T cells provided by a client to see the ability of their cells to kill. So this general outline and workflow can be adopted and adapted to a lot of different types of therapies we've done a number of them. Also, instead of just a 375 spheroid formation, we've also worked with primary tumor tissue as well. So really adjustable. Again, the benefit of that customizable assay is that you can tailor it exactly to what you need to answer the questions that you need. And the final one I'll go over here before delving into cell painting is adipogenesis, because this one seems to be picking up popularity lately with some of our clients and questions that they have. A lot of drug development campaigns are looking at either limiting or promoting adipogenesis. Excessive adipogenesis can lead to a lot of different problems like metabolic syndrome, type 2 diabetes, obesity. But on the other hand, some regulating this process in general is important because we do need it. It is necessary because adipogenesis helps to sequester the lipids in the body to avoid lipid toxicity from overaccumulation in different tissues like the liver or the heart. So the general assay outline for this project here that we did was to measure the ability of a client's test article, a compound, to promote adipogenesis using primary human subcutaneous pre-adipocytes. And our palette here would be hooked for nuclei. BODIPI is our stain for detecting lipid droplets, and again, cell mask for looking at overall cell morphology, so we know the boundaries of the cell. The method here is we seeded the pre-adipocytes and cultured them for 3 days before switching them over. We added the treatments and then switched them over to our differentiation media and followed our differentiation protocol. And we carried out this for about 12 days. And on the 12th day, we fixed and stained them and quantified their lipid droplet formation. The controls we had here were rosiglitazone. That's a medication that's used for type 2 diabetes, and it activates adipocyte differentiation. So that's our positive control for differentiation. We also had TNF alpha, which inhibits adipocyte differentiation. So that's our control for inhibiting the process. And then we ran these alongside of our client's test article. So you'll see from our representative image here, the second column is the BODIPI, the lipid staining. And you'll see that the test article was pretty comparable to rosiglitazone, but we can look at that further by actually quantifying. Again, there are a lot of different metrics here. I just focus on 2 of the more simple, robust ones, which would be the total area of lipid droplets that was formed in the cells, as well as the number of lipid droplets that were formed in the adipocytes. So in this case, you can see that rosiglitazone and TNF alpha did what we expected them to do. TNF alpha inhibited, rosiglitazone promoted relative to vehicle control or cells only control. And in this case, the test article's effect was pretty comparable to that of rosiglitazone, and it helped promote adipogenesis relative to vehicles. So this project, so this staining with BODIPIs, we've gotten a lot more asks about, especially in the CNS space. Of interest now to a lot of our clients is the role of lipids and lipid content in the CNS because it's becoming implicated also in contributing to the progression of neurodegenerative disorders, diseases. Accumulation of these lipids is associated with aging, inflammation, oxidative stress, which are all hallmarks of neurodegenerative diseases that we've known about for decades. So pretty interesting that we can take this one dye and apply it to so many different areas, different diseases, different cell types to answer different questions. Such as maybe even fatty liver disease models, and create different models to test our clients' different test articles for different disease areas. So the last thing I'll go over real quickly is cell painting. We have actually published a white paper earlier this year with Dave Swinney, who's one of the fathers of phenotypic profiling. So it kind of goes into the power of cell painting, the power of phenotypic profiling. So you can download that as well from our handouts or our website if you want to take a look at that. We also have this case study and some other information specifically about cell painting on our website as well. But for those that aren't familiar with cell painting or heard of it, but want to know a little bit more, I'll do a quick overview. It was originally designed as an unbiased method that was used in RNAi screens at the Broad Institute, and it's a scalable fluorescent and imaging-based approach that has now been adapted to drug discovery. Some of the most common use cases for it are for determining a mechanism of action of a compound that might work, but they don't know how it works, or target identification, identifying new targets that can be used in drug discovery. So the idea is you get a whole cell readout in response to a certain treatment paradigm. So it generates really unbiased information in this way, and you don't need to have any prior knowledge of the MOA or the safety profile because of this unbiased nature. So once you treat a certain cell type with a certain treatment paradigm, a certain compound for a certain amount of time, et cetera, what happens is you get a very unique signature, and we can extract over 1,000 features, like 1,000 to 1,500 different phenotypic features from a specific treatment paradigm. And that will kind of serve as a phenotypic, what we call a fingerprint, that identifies that cell that has undergone that specific treatment paradigm. And that you can use A549 cells, U2OS cells, simple cells, or take it all the way into iPSC-derived hepatocytes, cardiomyocytes, neurons, any type of cell type that is expandable into this assay. And you get back these rich data sets because you extract so many thousands of features. And then what you're really limited to is just your reference library here. So the typical palette is up at the top right in that chart. On the left column, you'll have the organelle or structure that is targeted by each of the dyes. And this is the canonical one that most people will go with, but we have had clients that come to us wanting to test out if a new palette would be, if a different palette that is more suited to what their needs or interests are, can be run in these assays. And actually one of them has been Bo-Dippy. So people that are interested in fat content, they'll replace one of the green dyes with Bo-Dippy and use that as their palette. So when we run these assays, our clients can choose their reference library. So for instance, if they're interested in kinases, if they think that their test article targets kinases, they can choose a library of kinase inhibitors, for example, and run their test article alongside that and try to map the fingerprints that their test article induces to those that are induced by the various inhibitors in that library. So essentially, the richer your reference library is, the more data points you have, the more information that you can get back about your test article. We have clients that are in the AI biotech space that like to use these rich data sets for helping them to develop and promote their predictive technologies. And so this is, I think that's why probably a lot of people are getting more interested in working with us in cell painting assays. This is a little bit of a simplified way of looking at how we do image analysis in these cell painting experiments. So across the top of each of these pictures, you'll have the stain, and then in the right under the name of the stain, you'll have the what we're gating for, what we're looking at. So this is how we start extracting metrics like number of spots, intensity of spots, fibers, textural readings, and the values, the quantitative values of each of these metrics vary with each of these treatment paradigms. So combinations of these thousands of metrics and values are what yield that unique phenotypic signature that we use to compare and identify targets or MOAs. So this is our example of one that we did in hepatocytes. These are iPSC-derived human hepatocytes in cell painting. So this was using the typical classic palettes. And here we have on the top row vehicle treated, and on the bottom we have cytokeratin D treated. And you can see just by eye how different these phenotypes are. And if you zoom in, for instance, you can see even more clearly how different these phenotypes are. And this is just what we can detect by eye. So using our analysis and our in our pipeline, we can extract way more than is visible to our eyes. And this is just one example of one concentration of one compound from a library that we tested. So essentially you can do this, run this assay, run these treatment paradigms in the entire reference library, and then we represent the data in a couple different ways. These are the most common ones that we use. On the left, we have a UMAP, so a projection of the data set, and on the right, you have a heat map and kind of a ranking system of those different compounds. So in the UMAP, you're looking for clusters. The more closely clustered your different treatments are, the more phenotypically similar those their fingerprints are. So for instance, baphomethycin A, way in the top left corner, is going to be very, very different from at the bottom, the podophylotoxin. And those, that big cluster that around the negatives, or that are categorized as the negatives here, they're pretty similar, and they probably don't induce much of a difference that we're able to capture. And this is just represented, the same data can be represented in a heat map here, where you can look at the correlation coefficient between those different fingerprints. So the lighter the color, the more similar they are. The darker the color, the more different. The more different that they are. And then you can use this to have kind of a ranking system of how different these the treatments, the phenotypic profiles are that these treatments induce. So you can figure out with those ranked test articles, or with the ranked reference library, where your test article falls. And if it falls closely or clusters closely with another compound that's within that library, it may be that they operate in very similar ways through similar pathways or mechanisms of action. So that's just kind of a smattering of some of the projects we've done. We've had so many different projects throughout the years, a lot of different interesting ones. And we're always excited to learn about the newest things that different companies are working on, the questions that are on their minds. So hopefully, if you have any questions, just drop them in the box and I'll get to them if I can. Otherwise, feel free to contact us, email us, visit our website for more information. So thanks for that. I'm going to hand it over now to Adele. She's going to talk about our digital storefront that we launched last year, where you can place your orders online to make your whole process easier for getting in touch with us and for getting for accessing us to discuss your project needs.
Thanks, Anna, for that very engaging overview. And hi, everyone. My name is Adele, Associate Director of Supplier Relations here at Scientist.com. And we've collaborated with Anna and the PhenoVista team to create an easier and faster ordering platform for the custom assay services and cell painting services that you just learned about. I'd like to take a few moments just to briefly cover how researchers can collaborate with the PhenoVista team easier and faster than ever before on this new online e-commerce platform, which was custom built and powered by Scientist.com. So if I'm able to share my screen, take you through a brief demo. So as you can see, this workflow really fundamentally begins at PhenoVista's website. To access this portal, simply click on Store here, where you will be taken to the homepage. We've designed this essentially for researchers to review all of the key information about the PhenoVista team, as well as their capabilities. You'll see some of the featured services Anna just spoke about, highlighted here. Customized it with them to essentially encapsulate all the key information. As you can see, you can search by specific keyword for what you're looking for. Maybe you know exactly what you'd like to collaborate with PhenoVista on. It's a very targeted opportunity. Alternatively, we built out a browse page for a more exploratory search. So if we jump to this page here, we designed this for PhenoVista to effectively highlight their entire services portfolio so you can find and identify what you're looking for, but also understand the full scope of PhenoVista's offerings and really rich, detail-oriented data and intake forms for each of these capabilities, essentially to drive more efficiency in those conversations with, Anna and her team or whoever you're connecting with. If we jump to one of these, we can show you an example of what that looks like. I'm going to select cell painting just because that's where Anna left off. But essentially, this allows you to really quickly and effectively articulate your specific needs when it comes to this project. So these custom forms are designed to capture as much information as possible, really allowing you to more clearly articulate the needs of what you're looking to collaborate on, but it also empowers Anna and her colleagues with a better way of kind of giving you that accurate pricing and quoting information faster and easier than a more general form. So essentially capture as much detail as we can there. You'll sign in and indicate where you're operating from, as well as any billing information. If you have a proposal, maybe you have a really, pressing deadline on when you need this by. Giving that information to PhenoVista upfront allows for better kind of prioritization. Once you complete the information on that form and initiate that request, it'll go to the PhenoVista team, who is able to quickly turn around with a message, set up a call, and that way, with all the information provided in this form, show up to those more meaningful discovery discussions. At that point, you'll be able to track this one request as well as any multiple. So we created this dashboard. Of course, mine is empty at the moment, but you'll be able to monitor the status and any updates for any ongoing projects that you have with PhenoVista. So everything is really consolidated. That way, instead of navigating multiple email threads, everything, including the proposal, any invoices, payment information, project details, are consolidated and organized by specific project. So this was something that we really wanted to prioritize with PhenoVista, and kind of enhancing that customer support and visibility into real-time updates here. One additional new update that we've kind of recently launched since partnering with PhenoVista is actually a custom AI chatbot that is in implementation for any new or existing e-commerce solution that we have. Very similar to the one actually that exists on our homepage. So additional way to engage with the team to better understand and nurture those conversations. Not, of course, to eliminate the human aspect of such highly complex and custom services, but really just streamline the process for both sides of that conversation. With all that said, I'll jump back to the homepage here, but this is a highly custom solution to support a variety of suppliers, capabilities, and sales cycles. As you can see with PhenoVista, very sophisticated, customized offerings. And we're really here to partner with anyone who has similarly very complex services that would like to, improve the website and the vision for their online sales. So if you're interested in kind of learning more about how to connect with PhenoVista on their storefront, or maybe discussing a storefront for yourself, please feel free to reach out to myself directly at adele@scientist.com or my colleague Andrew at scientist.com. We'll have our information linked, and we can find a time to connect. But with all that said, please feel free to send in any questions in the Q&A box, and we'll address as many as we can in the Q&A time, or either follow up with you directly via email. So thank you again. And with that, I'll pass it over to Liam.
All right, fantastic. Well, thanks so much, Adele, and for the great presentations. And with that, move right on to the Q&A session. And as a reminder, if you do have a question that you'd like to ask, you could submit it using the Q&A tab. All right, so first question of the day. So when thinking about the synaptogenesis assay, this person said that they saw you had low, medium, and high density seeding conditions. Do you know what those densities were approximately? Just curious to see whether they're compatible with some other assays we use.
Yeah, thanks for that question. I don't know offhand exactly what the densities were. It does change. It depends on the vendor. It depends on sometimes the time scale. It depends on the project. For synaptogenesis, we always run a few different concentrations and ratios of cells if it's a co-culture to run these assays. So no, not off the top of my head. And it kind of changes with each project that we do.
Fantastic, thanks. All right, Adele, one for you. And I know you mentioned this a little bit towards the end of your presentation, but one of the attendees here said that would this be a good fit for their business if they offer very custom services or off-the-shelf services?
Yeah, great question. Thanks, Liam. So our solution is designed specifically to promote and sell highly custom services, although as this configurable depending on what you offer. We'd love to discuss anyone's preferences or portfolio in more detail. Essentially, through extensive research and hundreds of conversations with both providers and buyers of these complex services, the gap in efficiency that we identified was really many websites still rely on the basic contact us form or kind of general intake form for these highly complex boutique service offerings, such as PhenoVista's custom assays. So by integrating a custom storefront and even a trained AI sales chatbot to nurture and streamline conversations, the PhenoVista team and other teams who are interested are able to streamline lead qualification and conversion without losing that personal human touch that's critical for services of this nature. So as a result, our goal for PhenoVista and anyone else is so that their sales teams can focus on high quality, market qualified leads while still co-creating with clients on these personal projects. So accelerating your discovery process, empowering both sides of the platform.
Perfect. Thanks, Adele. All right, next one here. Can you elaborate on how you work with us? So, and I think this question will be for you. Can you elaborate on how you work with other service providers? For example, if we would like both imaging and RNA-seq data, would we also need to contact the RNA-seq company?
Yeah, good question. We've been getting more and more of those types of asks. No, you don't. So our hope is, as part of our excellent customer service, we will contact them for you. We do have partnerships with companies that offer these orthogonal assays. So we can provide more of like a one-stop shop deal. And then when we send you a proposal and quote and estimates, it already includes the costs of all those other assays as well. So of course, if you have a particular company that you want to go with, we're happy to also contact them and work it into our workflow. But we do have our own service providers that we prefer to go to. But as with anything that we're offering on our assays, it's customizable.
Perfect. Thanks a lot. All right, next one here. So this one is for you, Adele. What would be the next steps to get started with a web store like this?
Yeah, next steps are very simple and tailored to the needs of the company. So first, we'd schedule a 30-minute demo where we could dive into your specific features, capabilities, provide the most value for your business. So during the demo, we'd explore kind of your current lead conversion processes, identify opportunities to improve this, and demo how our solution could help your team focus on these market qualified leads. So very customizable, and the best way is just to get in touch with our team directly.
Perfect. All right. Thanks a lot. And I think in the interest of time, we'll just make this next question the last one. So on it, do you provide images and raw data for clients to make our own graphs or do their own analyses?
Yeah, we definitely have that capability, as simple as shipping a hard drive that we have with images or whatever you need. We also have an extranet service for smaller files. So we can just deposit the files for you to download the raw Excel files or whatever format you need. Some clients like to take our images and run it through their own pipelines for analysis, just to compare their pipelines with ours. And that's a very common one we get in our image analysis services. So yeah, that's definitely an option. Whatever we generate in our lab, the data is yours. So yeah, absolutely accessible.
Excellent. All right. Thanks so much. And yeah, thanks so much again, Anna and Adele, for the really fantastic presentations. It's really been a pleasure having you with us.
Thank you.
Thanks, everyone.
And in closing, thanks again for attending the Scientist.com and PhenoVista webinar, and we'll see you again next time. Have a great day, everyone.